Close your eyes. Beneath your skin, far below thought or breath or sound, your body is reading. It is reading a story written in a language so old it predates memory, predates speech, predates even the first whispered names for light and shadow. This story is not written on pages, but on strands, tiny spirals of code tucked within every cell of you. Each of your trillions of cells carries a version of this story. It's written in a script called DNA, deoxyribonucleic acid, if you feel like wrapping your tongue around the whole name. But you don't need to. Tonight, we will call it what it is, your blueprint, a quiet, twisting staircase of letters, curled like vines in the dark. Imagine it now, a double helix spinning slowly in the warmth of your inner world. It is as if two ribbons dance in a spiral, holding hands through invisible bonds, cradling the secrets of who you are. They are made of 4 small letters, A, T, C, and G, like the notes of a lullaby. A always pairs with T, C always finds G, like soulmates in molecular form. These letters form words. The words form instructions. Some of these instructions tell your heart how to beat, your skin how to stretch, your eyes how to curve light into sight. Others are quieter, tucked in corners, rarely read, waiting perhaps for a moment when the body calls for them. This is not just a story about you. It is the story of life itself, passed down from every ancestor who ever breathed. Your DNA is so long, it could stretch to the moon and back, multiple times if laid end to end, and yet it coils so tightly, so elegantly, it fits inside the microscopic center of each cell. It is the most compact, most efficient information system we know, the original archive of nature. Think of it like this. Your body is a great library. Each cell is a room in that library. Inside each room is a set of encyclopedias, exact copies. These are your chromosomes, 46 volumes, arranged in pairs, containing everything you need to grow, heal, live, and become. When your body needs to do something, build a muscle fiber, create a hormone, replace a skin cell, it opens a chapter in the DNA, copies the needed sentence, and sends it out to be followed like a recipe. This sentence becomes a protein, the tiny machines that make you, you. Proteins are the builders, the movers, the signalers. Some carry oxygen, some break down food, some transmit thoughts. Each one begins as a line of DNA, faithfully transcribed into action. Even as you lie still, even as you drift towards sleep, your cells are busy reading and building. Now let's go even deeper. If DNA is the book, then the cell's nucleus is the reading room, quiet, protected, lit by the warm hum of life. Here, molecular scribes called enzymes move along the strands, unzipping the helix, copying one strand into a message made of RNA, a temporary note passed from the quiet archive to the bustling factory floor. This RNA carries the instruction outward to the cytoplasm, the cell's open space. There, ribosomes, tiny, humming machines, read the message and begin to build. One amino acid at a time, they string together the chain, folding it into a precise 3-dimensional tool. You don't feel this happening, but it's happening, now and now and now again. Every cell of yours knows this process by heart. From the top of your scalp to the tips of your toes, they are fluent in this language you never learned in school and yet have always known. And what's more beautiful, the story is not silent. It changes, sometimes by nature, sometimes by accident, sometimes, now, by choice. A small shift in one letter, a single swapped base can change a protein. It can alter a trait. It can cause a disease. It can make a heart beat differently, or cause a blood cell to curve when it should be round. We call these small shifts mutations. Some are harmless, some are life-threatening, but all are part of the ongoing conversation between nature and time. In this code, ancient, imperfect, exquisite, lies the possibility of healing. What if we could go in and gently correct a single letter? What if we could uncurve a sickled cell? What if we could listen to the blueprint and help it rewrite itself? These are not dreams anymore. They are questions being answered in laboratories all around the world, with tools that come from nature itself, with methods precise enough to change a single base without disturbing the rest. But before we can explore those tools, before we speak the word CRISPR, you must understand the magnitude of the script we're touching. This is not just biology, this is authorship. And the genome, your genome, is not a static page. It is a living manuscript. You're not made of solid stone, you're made of letters, constantly read and rewritten, folded into your breath, your bones, your dreams. You're a story in motion, a song softly sung in the language of the life. As you lie here now, your cells continue to hum their instructions. You do not command them, but they obey a wisdom deep inside. This is the blueprint within, quiet, hidden, powerful. And though it is invisible, it shapes everything you feel, everything you are. The question then is not whether the blueprint is beautiful. It is. The question is, what happens when it goes awry? What happens when a single letter misguides the melody? Let's drift next into that question, into the soft shadow where illness begins and where, perhaps, healing can follow. And yet, like any great story, your script is not without its imperfections. Inside the folds of your DNA, that elegant helix humming quietly inside every cell. Small changes sometimes slip in, tiny edits, little echoes of chance. Imagine the blueprint you carry not as flawless marble, but as parchment, ancient and well used. Pages, handled gently by time, but not always perfectly preserved. A single letter, miscopied. A page folded at the corner. A comma placed where a period should be. In the language of your DNA, these typos are called mutations. A change in a single base. Perhaps an A becomes a T. Perhaps a sentence no longer reads clearly, or a word vanishes entirely. Most of the time, these changes do little. The cell keeps reading, the music plays on. But sometimes, just sometimes, a small mutation changes everything. The way a slipped note changes the meaning of a song, or the way one cracked brick can shift the strength of a whole bridge. Consider this. Some people are born with a change in a gene that makes hemoglobin, the protein in red blood cells that carries oxygen. The blueprint tells the body how to fold it just right, but when the code is altered, the shape changes. Instead of round, the cells become curved like sickles, jagged, stiff, and prone to getting stuck. This is called sickle cell disease, not because the body fails, but because the instructions were slightly misread, and the body, faithful as ever, followed them exactly. In cystic fibrosis, another mutation affects a different protein, one that moves salt in and out of cells. With the wrong instructions, the salt gets stuck, and thick mucus builds in the lungs, making each breath harder than the last. These are just two stories in a vast complex library of genetic conditions, each caused by a single line, a misplaced letter, a silenced word. And yet, for those who live with them, the effects are profound, touching every heartbeat, every movement, every day. Still, these mutations are not punishments. They are part of the natural language of life. Errors that slip in not from negligence, but from the sheer scale of the copying task. Your body is duplicating DNA all the time, millions of letters, again and again. It is remarkably accurate, but not perfect, and evolution too is built on change, on trial and error, on what survives and what fades. Some mutations lead to new traits, new advantages. They are the quiet engines of diversity, of adaptation, of life's resilience. Others, though, bring pain, limit, or loss. When these mutations are inherited, passed from parent to child, they become part of the family script. The blueprint is shared, not chosen. And so, some families carry the weight of diseases passed through generations, not as a fault, but as a legacy written into the cells. For centuries, medicine could only observe. It could describe the symptoms, try to ease the pain. But it could not reach the root of the story, the DNA itself. It was like trying to fix a novel without ever seeing the manuscript. But now, slowly, tenderly, we are learning to look directly at the source, to read the code, to understand it, to see the typos for what they are, not curses, but solvable puzzles. And this is where something quietly extraordinary begins. Because if the disease is caused by a single misplaced letter, what if we could correct it? not mask it, not suppress it, but go to the source and edit the blueprint itself? It sounds like the work of dreams, but dreams, when coupled with science, become experiments, and experiments, when repeated with care, become therapies. The body is still sacred. It is still mystery and miracle, but we are starting to learn its language, and within that language there may be a way to heal. Some might say we are rewriting nature. Others say we are listening more closely to what it has always told us, that life is editable, that stories can change, that healing sometimes begins with reading more carefully. But how do you edit a molecule? How do you find a single letter in a library of billions? How do you change it without harming what surrounds it? That is where the next chapter begins, in the salt pans of Spain, in the study of the smallest life forms, bacteria defending themselves with memories made of DNA. Out of that strange and ancient defense system came a discovery so quiet, so small, yet so precise, it changed everything. Tonight, let your breath soften, let your body be still, and as you drift towards sleep, hold the question gently. If nature made an error, could nature also hold the cure? In the bright salt flats of southern Spain, beneath skies that shimmer white with silence, scientists studied some of the smallest life on Earth. Bacteria, invisible threads floating in ancient brine. To most eyes, they were unremarkable, simple, single-celled, countless. But within them was a secret, a pattern unlike anything seen before. It started as a curiosity, rows of repeating sequences in their DNA, like mirrors echoing each other. Between the repeats, strange fragments, like clippings from a magazine pasted into the same place, over and over again. They called these sections CRISPR, clustered regularly interspaced short palindromic repeats. The name is long, complex, but the idea is beautiful in its simplicity. These fragments, it turned out, were memories, not memories like yours, not smells or songs, but molecular records of past invaders, viruses that had once tried to harm the cell. The bacteria had captured pieces of the enemy's DNA and stored them, like trophies or wanted posters, each one a warning: this sequence tried to hurt us, watch for it. When that virus returned, the bacteria were ready. They transcribed the CRISPR sequence into a guide, a strand of RNA, and handed it to a small protein, shaped like a pair of scissors. That protein was called Cas9, and together, CRISPR and Cas9 became a defense team, one carrying the memory, the other ready to cut. When the matching invader appeared again, the guide RNA would recognize it. Cas9 would slice it. The danger would be gone, cleanly, precisely, quietly. This was nature's immune system for bacteria, an elegant way to remember and remove threats. But to researchers watching from the outside, it sparked a different idea. If this system could find a specific piece of DNA and cut it, then maybe, just maybe, it could be used in other cells too. Human cells, animal cells, any cell with a genome. It was a whisper of possibility, a natural tool millions of years old suddenly seen not as just a defense, but as a way to edit life. The shift began slowly. Scientists began borrowing the CRISPR system, rebuilding it in labs, simplifying it for precision. Instead of letting bacteria carry their own guides, they designed their own, short RNA strands written by hand, aimed like arrows at a chosen genetic target. It worked. A guide RNA found its matching sequence. Cas9 followed. The DNA was cut, exactly where the scientist wanted. It was like giving the body a scalpel made of light. No more guessing, no more imprecise trial and error. For the first time, we could go into the genome with purpose and change one tiny sentence without disturbing the paragraph. CRISPR became the simplest, cheapest, most precise genetic tool ever discovered, and it came from salt-dwelling microbes, carrying memories in their code. This is how science often moves, a leap born of patience, a revolution born of curiosity. The CRISPR system had always been there, but only when someone listened closely did its full power reveal itself. Now, imagine what that power means. A child born with a faulty gene could, one day, receive a treatment that snips the mutation out and repairs it with something healthy. A disease long considered incurable might quietly vanish, not with loud fanfare but with a whisper inside the cell. CRISPR is not magic, but it feels like a kind of grace, a way to correct without punishment, to heal without forgetting. Still, it is young, barely a decade old in human hands, and like all powerful tools, it carries weight. To edit the genome is to touch the essence of a person, to alter the text that makes them, not just their illness, but potentially their traits, their futures, their children. And so CRISPR brings with it more than precision. It brings responsibility. But for now, let's linger on the wonder. The way bacteria, in their fight to survive, gave us a gift, a tool that lets us step into our own blueprint, not as victims of its flaws, but as caretakers of its possibility. You might imagine CRISPR like a gardener's tool. It can prune away damaged growth, encourage new shoots, reshape the landscape gently, with intention. Or you might see it as an editor's pen, a red ink line through a harmful phrase, a corrected word written in its place, not rewriting the whole book, just making sure the story reads clearly again. The discovery of CRISPR was not loud. It happened in journals and late-night labs, in petri dishes and careful notebooks. But its echoes are loud. Echoes that reach into hospitals, into families, into futures. And all of it began in something as small as a bacterium, something we cannot see, but whose secrets are now rewriting what we can be. As you rest, think of the scale of that shift, the way vast possibility bloomed from the smallest corner of the natural world, and how now, with a guide and a pair of scissors, we begin to imagine healing not just through medicine, but through the language of life itself. Yet for CRISPR to do its work, it must find the exact place to cut. Out of billions of letters, how does it know where to go? Let us drift next into that mystery, into the quiet elegance of how CRISPR finds its way. Inside your cells, where silence becomes structure, CRISPR moves like a traveller with a map. It does not guess. It does not wander. It searches, methodically, faithfully, for one destination written in the code. You carry within you 3 billion letters of DNA, a sequence vast beyond imagining, folded and packed into the quiet of each nucleus. In this infinite novel of life, CRISPR must find a single sentence, a single typo, a place where something has gone awry. How does it know where to go? The answer begins with a guide, a small strand of RNA, no more than 20 letters long. It is called the guide RNA, and it holds a memory, a mirror of the sequence to be found. This guide is handcrafted by scientists, shaped to match the faulty part of the gene, a perfect puzzle piece to a problem in the body. Once created, this RNA guide is bound to Cas9, the protein scissors we met before. Together they become a search team, one leading, the other ready to act. Now picture them floating through the cell's nucleus, drifting gently among coiled DNA strands, testing each section they pass. A, T, C, G. Does this match? Number, and on they go. Billions of letters, and yet the guide knows because the shape of the sequence it seeks is precise, as exact as a lock to a key. When the match is found, something incredible happens. The RNA guide hugs the DNA strand. It wraps around its target like a vine around a branch, a gentle recognition. I know you. And then Cas9, the scissors, come alive. They open, then close, snipping both strands of the DNA with care. Not a violent cut, a deliberate pause in the text. It is this break, this controlled incision, that begins the healing. DNA, like skin, responds to cuts by trying to mend. When it senses the break, it brings tools of its own. Repair enzymes gather, and the cell begins to stitch the strand back together. Sometimes this mending introduces a mistake, a tiny deletion, which can disrupt a faulty gene. Other times, scientists provide a repair template, a clean version of the sentence which the cell copies into place. It's like handing the cell a sticky note that says, "Try this instead." In either case, the change is made at the source, the gene is altered, the blueprint is corrected, and the body, following its quiet instructions, responds. This entire process happens in a space smaller than a grain of dust. It takes seconds, and yet it holds the weight of centuries. A new way of healing, guided by design, not chance. Still, this precision has limits. The guide RNA must be accurate. Even a single mismatch can confuse the process. An error might cause Cas9 to cut the wrong place, an off-target effect as scientists call it. To prevent this, immense care is taken. Computers simulate the genome, checking for near matches. Experiments test and retest. Like an artist perfecting a brushstroke, the guide RNA is refined until it leads only to the place it's meant to go. Think of CRISPR as a kind of molecular GPS, but instead of streets, it navigates chromosomes. Instead of buildings, it passes genes. And when it reaches the faulty one, it doesn't demolish it. It gently rewrites the address. The elegance of this is hard to overstate. Before CRISPR, gene editing was like carving a sculpture with a hammer. Now it's more like writing calligraphy in wind, precise, fluid, softly powerful. This is what allows CRISPR to treat real conditions, not in theory, but in real people. Diseases caused by a single broken gene can now be addressed directly, with a tool that finds only that gene and nothing else. This kind of specificity is what makes CRISPR special. It doesn't flood the body, it doesn't overwhelm the system, it moves with intention, changing only what needs to change. And because it's so targeted, its effects can be permanent. One edit, one repair, a gene corrected not just for now, but for the rest of a person's life. Of course, the journey isn't always easy. Getting CRISPR into the right cells is a challenge. Sometimes viruses are used, emptied of their own danger, and filled instead with healing. These delivery methods are being improved all the time. Newer, safer vehicles, sharper guides, more elegant versions of Cas9. This is a field still evolving like the DNA it edits. But already there are stories of success. People with blood disorders, treated once, and freed from the symptoms that shape their lives. Imagine carrying a mutation all your life, then one day, after a single treatment, feeling your cells breathe differently, as if your body has been waiting for this moment all along. That's the promise of precision, to touch just one word in a sentence and change the meaning from suffering to relief. CRISPR doesn't just find its way, it knows the way, because it is built from memory, from mirrors, from a guide that recognizes the place it belongs. As you lie still now, imagine those tiny molecules at work, a guide in one hand, a gentle blade in the other, moving through darkness with perfect clarity. They seek not to change who you are, but to return you to a version of yourself before the error appeared. And what happens when this power is directed at a real disease, when the fault in the blood is found and gently repaired? Next, we will walk with the story of blood itself and see how CRISPR turns a legacy of pain into the beginning of relief. Your blood is not still. It travels endlessly, looping through your body in silent rhythm, carrying warmth, oxygen, and memory through vessels as fine as threads. With every heartbeat, red cells deliver breath to your tissues. They cradle oxygen in iron-rich arms, flowing smoothly, swiftly, with practiced grace. Their shape, round and soft like tiny pillows, allows them to bend, to twist, to slip through narrow capillaries without losing form. But for some, this story is different. A single change in the DNA, one letter in one gene, reshapes the structure of hemoglobin, the protein that carries oxygen inside red cells. This tiny change bends the red cells into crescent shapes, like sickles, sharp and stiff. Instead of flowing, they stick. Instead of lasting, they break down too soon. This is sickle cell disease, and it begins not with an injury or infection, but with a blueprint altered in one line, a mutation passed quietly from parent to child. The consequences are anything but quiet. Pain comes in waves, deep, aching episodes known as crises. Organs suffer from lack of oxygen. Fatigue becomes a daily companion, and in many parts of the world, children with sickle cell disease live shortened lives. It is a heavy inheritance, but it is also an invitation to ask, can this story be rewritten? This is where CRISPR enters, not with drama or magic, but with focus. To understand how CRISPR helps, you must first meet a gene called BCL11A. It doesn't cause sickle cell directly. Instead, it silences something else, a fetal gene, one you used before you were born. That fetal gene makes a form of hemoglobin that isn't affected by the sickle mutation. In babies, it carries oxygen beautifully. But soon after birth, BCL11A turns it off, like a light switch, dimming the old to make way for the adult form. In people with sickle cell disease, this adult form malfunctions. But the fetal version still works. It just lies dormant, waiting. So scientists had an idea. What if CRISPR could silence BCL11A? Not destroy it, just turn it down, removing the block on fetal hemoglobin. The approach is simple in concept, complex in execution. First, blood stem cells are taken from the patient's body. These are the cells that live deep in the bone marrow, generating all new blood. In the lab, CRISPR is introduced. It targets a precise region in the BCL11A gene, snipping it in a way that disrupts its ability to silence. Then, the edited stem cells are returned to the patient, infused gently, like a transfusion. From these few altered seeds, a whole new blood system begins to grow. And what emerges is remarkable. Cells that produce fetal hemoglobin once again. Red cells that remain round, soft, flowing. Oxygen carried without obstruction. Pain silenced where once it roared. This therapy is already real. Clinical trials have treated people who lived with chronic pain, frequent hospitalizations, and the weight of inherited suffering. Some of them now report a life they never imagined. One with no more crises, no more limitations, a life where their own cells no longer betray them. It is not a cure in the simplest sense, but it is a profound rewriting, not of the past, but of what the future may hold. Of course, there are challenges. The treatment is complex. It involves chemotherapy to make space for the edited cells. It requires time, monitoring, precision. And access is uneven, available in some countries, still out of reach in others. Cost and infrastructure remain hurdles, but the path is visible now, and each step forward brings more light. There are other approaches too. Some scientists use CRISPR to directly fix the sickle mutation itself, repairing the faulty hemoglobin gene at the source. Both strategies hold promise. Both reflect a future where sickle cell disease is not feared, but treated, not endured, but unmade. And perhaps even more profound is what this therapy represents. A new kind of medicine, not made in factories, but made from within. Your own cells, edited with precision, become your medicine. Your healing rises from your blood, written into the marrow of who you are. This changes how we think about illness. No longer something only managed from outside, but something rewritten at its root. And in that rewriting, there is poetry, there is choice, there is the quiet possibility of release. As you rest now, imagine the journey of one red blood cell, round and whole, flowing through vessels without pain. It carries oxygen like a lantern in the dark, lighting the path to muscles, to brain, to breath. It is a messenger of what's possible. What if this was only the beginning? If CRISPR can restore blood to ease, might it one day bring light back to those who've lost it? In the next chapter, we will follow the science into the realm of sight, where genes shape vision, and CRISPR reaches into the retina to let the brain see again. Close your eyes for a moment. What you feel now, the softness of darkness, is something some people carry always. Not because their eyes are closed, but because something deep within those eyes no longer speaks to the light. The eye is not just a lens or a mirror, it is a translator. It gathers light, photons, and converts them into signals. It turns waves of brightness and shadow into pulses, then sends them along the optic nerve to the brain, where vision truly begins. It's not your eyes that see, it's your brain, painting pictures from electrical whispers. But those whispers begin with cells in the retina, thin and delicate like lace. And when a single gene in those cells is broken, that whole whispering process falls silent. In one inherited condition called Leber congenital amaurosis, or LCA, a mutation in a gene called CEP290 means the cells in the retina cannot function. They're present, physically there, but unable to do their job. The bridge between light and sight is broken. Children born with this condition often lose vision early, some in infancy. They may see only shadows, or light without shape, and because the disease is genetic, the cause lies not in trauma or age, but in the blueprint, the DNA. But here, once again, CRISPR offers a quiet, careful intervention. This time, scientists don't take cells out of the body to edit them. Instead, they bring the editing tool into the eye. A single injection beneath the retina. Inside, a harmless virus carries the CRISPR components, the guide RNA, precise to the faulty sequence, and the Cas9 enzyme, the molecular scissors. This viral envelope delivers its message into the waiting cells. Then the guide finds its place. Cas9 makes the cut, and the gene, broken, misshapen, is gently corrected. It's like lifting a veil from the page, restoring a line in a forgotten stanza of vision. This is called in vivo editing, editing within the living body, not in a dish or under a microscope, but in the soft tissue of a human being. In early trials, some patients with LCA reported changes as small as they were profound. A flicker, a shadow moving, a new sense of brightness where there was none. One young man described seeing a doorway where before there had only been blackness. Another could make out light coming through a window, something they had never truly seen. These may seem small, but as someone who has never followed the shape of light, they are revelations. CRISPR did not give them new eyes. It reactivated cells that had waited in silence. It allowed the body to speak its own language again to complete the interrupted sentence of sight. And it did so with precision. In the dense, delicate fabric of the retina, there is no room for clumsy tools. Only something as specific as CRISPR could attempt this. As this therapy evolves, scientists hope to treat other causes of genetic blindness, mutations in different genes, each with its own unique disruption, each one a different loss signal, a line of code waiting to be restored. But just as with blood, these treatments are not yet common. They live in trials, in early stages. There are still questions to answer, timelines to understand, effects to monitor across time. And yet, for the first time, we are not only imagining a future without inherited blindness, we are beginning to build it. That is the power of editing not just words, but the instructions that shape experience. And it reminds us of something deeper, something woven through all of biology, the capacity to wait, to hold potential, even in silence, to be ready, even in the dark. For the retina to respond after years of blindness, those cells had to survive, dormant but intact. CRISPR was not resurrection, it was awakening. And you too are filled with dormant potential, genes that are silent, cells that remember older ways of being, a body that holds more stories than it tells. In treating blindness, we are not creating something new, we are gently returning to something lost. But if we can restore vision, if we can whisper light back into the brain, then what else might be possible? If we can edit one cell in one eye, what of the rest of the body? What of diseases that move more freely, touch more organs, hide deeper in our genes? This is the frontier CRISPR now approaches, a place where power and precision meet uncertainty. Next, we will drift into that space of care and caution, where science begins to ask, just because we can, should we? You lie still. Your body breathes softly, your cells, as always, move without command. Inside, quiet edits are taking place, not CRISPR, but natural change, proteins folding, enzymes acting, genes switching gently on and off like soft lamps in the dark. Your biology is not static, it has always been fluid, shifting, adjusting, evolving. But now, for the first time, human hands have tools to intervene with intention. And this is where the questions begin. CRISPR is extraordinary. It can remove a mutation with precision, activate silent genes, silence harmful ones, and rewrite tiny chapters of life's book. But it is not perfect. Like any powerful tool, it comes with risk, and with that risk, a deep responsibility. What if CRISPR cuts in the wrong place? What if the guide RNA misreads a sequence and changes a gene that should remain untouched? These are called off-target effects. Small accidents, sometimes invisible, sometimes serious. In some experiments, off-target cuts have been linked to unwanted mutations, even changes that could lead to cancer if left unnoticed. Researchers are improving the system every year, sharpening the guide, refining Cas9, creating newer versions like high-fidelity Cas9 to reduce these risks. Still, no system is without flaw. And even when CRISPR works exactly as intended, there are deeper questions. For instance, should we use it only to treat diseases? Or is there a line between therapy and enhancement? If we can remove a gene that causes blindness, should we also remove one that leads to nearsightedness? What about genes linked to lower intelligence or shorter stature, or other traits we find inconvenient? You begin to see the slope, gentle but slippery. It is one thing to remove suffering. It is another to edit for preference, to begin choosing the kind of person someone becomes before they ever take their first breath. This leads us into the realm of germline editing, when changes are made not to cells in the body, but to sperm, eggs, or embryos. These edits are not just personal, they are permanent. They are passed down to future generations, written into the family tree before it even grows. In 2018, a Chinese scientist announced he had edited the embryos of twin girls, removing a gene that allows HIV to enter cells. The girls were born. The edit worked, but the backlash was swift. Not because preventing HIV is wrong, but because the path taken was silent, unauthorized, and ethically unsteady. No one knew the long-term effects. No one could predict how these changes might ripple through the genome, through time, through the lives of children who did not consent to be edited. Science has power, but power without wisdom can become harm. And so, the world paused. International councils met. Laws were written. Conversations began in earnest. Where is the line? Who decides what should be changed? Who gets access to this power, and who is left behind? These questions do not have easy answers, and yet they must be asked. Because CRISPR is not just for rare diseases, its reach is expanding into cancer therapies, organ transplants, even agriculture and livestock. It is changing how we think about nature itself, no longer fixed but editable, no longer sacred by default but subject to design. So we ask, what does it mean to be human when our DNA can be edited like text? Do we lose something in that shift? Or do we gain something? Agency over suffering, choice over fate. There are no final answers tonight, only open questions, and the gentle reminder that science must walk with humility, even as it strides forward. For now, most gene editing is limited to somatic cells, the body's tissues, not its future. These edits affect only the person treated. They do not pass on. They are bounded, measured. And yet, even here, caution matters, because what we change in one part of the body can sometimes echo elsewhere. The body is not a machine with separate parts. It is a symphony, and changing one note may shift the whole melody. That is why each step with CRISPR is taken slowly, each trial carefully monitored, each patient followed closely across time. Science is not afraid of risk, but it is learning to honor it. And so, as you drift towards sleep, you rest in a moment of balance between what we can do and what we should. There is wonder here, but also weight. There is the promise of healing, but also the potential for misuse. And in this balance, you find the stillness of reflection. Not everything broken needs to be fixed. Not every difference is a defect. Not every possibility should become action. But some are worthy. Some edits restore life to the living. They return breath, blood, vision, movement. They ease pain that has echoed for generations. And those changes remind us what this is truly about. Not playing God, but listening more closely to the code already written and asking how gently it might be mended. Still, this power does not belong to one nation, one scientist, one voice. It is spreading through borders, through systems, through families and futures. Let us now drift outward, across oceans and continents, to explore how CRISPR is reshaping lives all over the world. The wind does not recognize borders, nor does DNA. It travels from mother to child, from breath to breath, unfolding in every body everywhere, no matter the flag that flies above. Sickle cell disease is not American. It is not African. It is human. So are blindness, cancer, muscular dystrophy, and countless other conditions rooted in the deepest corners of our cells. CRISPR, too, is a global language, a technology born from bacteria shaped by researchers from every continent and spoken now in labs across the world. But as with all things powerful, its reach is uneven. In some countries, gene editing is moving forward with careful urgency: clinical trials, biotech startups, university grants, patients enrolled, monitored, slowly healed. In others, the technology feels distant, a whisper of promise, barely heard above the noise of everyday need. For while DNA is shared by all, access to its editing is not. Consider the story of sickle cell disease again. It affects millions, but the highest concentration is in sub-Saharan Africa. And yet the first CRISPR-based therapies, costly, complex, and still evolving, have appeared primarily in the United States and Europe, where trials are funded, where hospitals are equipped, where gene sequencing is routine, and gene therapy is a frontier worth funding. In contrast, many clinics in low-income regions lack not only CRISPR, but even reliable access to blood transfusions and antibiotics. It is not science that fails, it is systems. It is the scaffolding around the cure: infrastructure, training, storage, policy, trust. And so, for now, CRISPR's miracles exist in pockets, shining islands in a wide and uneven sea. But even islands send out light. In India, scientists are developing low-cost CRISPR platforms to treat beta thalassemia, another inherited blood disorder that burdens rural families. In Brazil, researchers are exploring how to use gene editing to control mosquito populations, limiting the spread of Zika and dengue. In South Africa, ethical think tanks are shaping how CRISPR can be safely and fairly applied in communities with long histories of medical injustice. And in every corner of the world, young scientists are learning the language of genes, writing code not on computers, but in life itself. The technology is spreading, and as it spreads, it adapts. Some nations are cautious. Germany, for instance, restricts all forms of germline editing, fearing echoes of eugenics, carried forward through blood and memory. Others move faster. China invests heavily in gene editing trials, sometimes pushing boundaries, sometimes racing ahead of global consensus. There is no single pace, no single policy, only the question: what is the role of a cure in a world still marked by inequality? For a child born with a genetic disease in Ghana or Guatemala, the promise of CRISPR may seem as distant as a constellation, visible but unreachable. Yet even in these places, seeds are being planted. Gene therapy centers are emerging. Grants are funding local innovation. Collaborations between countries are taking shape. Networks of healing, not ownership. The science is becoming more open. Shared tools, shared data, shared ethics. Because the human genome is not national, it is collective. And as prices drop, as editing tools become more refined, more portable, more scalable, the hope is that CRISPR will not belong only to the wealthy few. But this shift must be intentional. It is not enough to have the cure. We must build the bridges that deliver it, train the hands that wield it, listen to the cultures it enters. Because gene editing is not just technical, it is deeply personal, deeply cultural. In some communities, illness is not viewed as a flaw, but as a part of identity. In others, decisions about medicine are made not by individuals but by families, by elders, by shared belief. CRISPR must be humble in these spaces. It must ask permission. It must be a guest, not a force. And always, it must be accompanied by education, by clear explanation, by consent that is full and free. This is the future of global medicine, not one model exported outward, but many models rising together. CRISPR may have begun in a lab, but its future lies in schools, clinics, homes, and conversations between doctor and patient, between scientist and society. And perhaps one day, a child born in a remote village, with a disease long seen as fate, will receive a therapy just as advanced as any offered in New York or Geneva. Not because charity arrived, but because equity did. This is the promise, not just of a genetic cure, but of a shared path to healing. As you rest now, imagine that healing stretching across the globe, not in flashes of brilliance, but in threads of connection. A CRISPR therapy developed in Boston used in Nairobi, a discovery made in Lagos guiding trials in London. Knowledge moving, gently and justly, the genome becoming not just editable, but accessible. Still, as we reach outward, we must also think forward, because some edits affect only one person, others echo into the future. And so next, we turn toward the idea of generational change, of not just healing the present, but preventing illness before it ever begins. There is a different kind of healing, one that does not begin with illness, but with possibility. Not a cure for someone already suffering, but a quiet prevention, woven into the code before the first breath is ever drawn. This is the dream of germline editing, a future where a single change in a single embryo ripples forward, not just for one life, but for every life that follows. Imagine it. A child is born without the mutation that shaped their family for generations. They grow, they live freely, and when they have children, the corrected gene passes quietly on. A new branch on the family tree, one without the weight. The thought is luminous, a kind of forever healing, a moment of intervention that rewrites not just a body, but a future. This is not science fiction. The tools exist, the maps are drawn, the CRISPR scissors are sharp enough and the guides are already in our hands. But this dream, it whispers as much caution as it does hope. Because germline editing changes everything. When we edit an embryo, we are not treating a patient, we are shaping a person who cannot speak yet, who cannot choose. We are writing into silence, hoping the words we choose will echo kindly in lives we cannot see. It is a different kind of power, one that requires deep humility. For many, the question is not just, can we do this? But should we? Some argue yes, that if we can prevent a disease, spare a child from suffering, then it is our moral responsibility to do so. Others pause, they worry about unintended effects, that a small well-meaning change might introduce new risks we don't yet understand. DNA is not a switch, it is a web. Tug on one strand, and others may move. And even when the science is sound, there are other risks. Social, emotional, cultural. Who decides what should be edited? Who draws the line between healing and enhancement? Between therapy and design? What begins with noble intentions could, unchecked, lead to something colder. To choosing eye color, height, intelligence, to the quiet pressure to be more perfect, less human. This is why, around the world, germline editing is either banned or heavily restricted. Global councils meet, ethics boards debate, scientists call for caution, for consensus, for time. And still, the dream lingers. Because in rare cases, there are families facing impossible choices, two parents who carry the same deadly mutation, with no way to have a child who would not inherit the disease. For them, germline editing is not enhancement. It is hope. It is the possibility of one child, healthy, whole, free from the shadow of inherited pain. In these spaces, the debate becomes tender, personal, real. No longer abstract philosophy, but the voice of a mother, the longing of a father, the question, what if we could stop this? Just this once. And so, the world listens. Research continues, slowly, carefully. Model organisms, mice, zebrafish, are studied to understand long-term effects. New tools emerge, like base editors and prime editors, offering gentler, more precise changes than Cas9 alone. And always, the ethical conversations walk alongside the science, like a lantern held low, lighting each step, but never rushing. Because the goal, is not to race ahead. It is to walk together, to build a future where healing is shared, not forced, where prevention is offered, not imposed, where every change is made not from fear, but from love. And perhaps, one day, a small team of scientists and parents will sit together, reading the DNA of an embryo not yet born. They will see a mutation, one that has taken lives, and they will have the chance to change it. Not to make the child better, but to spare them from the worst. A single letter swapped, a life changed. And in the stillness that follows, they will know this was not power, it was protection. This was not design, it was care. And the child will grow and never know what they were spared, and their children may grow too and never carry that shadow. It is not immortality, it is not perfection. It is the quietest kind of miracle, a disease that ends in a single generation and does not return. As you rest now, imagine those future children, not chosen, not sculpted, just free. Free to live without the script that once seemed unchangeable. Free to write new stories with bodies that do not carry inherited harm. That is the dream of forever healing, and like all dreams, it is delicate. We must carry it gently. And yet, as we close this journey, you may wonder, what comes next? CRISPR is still young. There are new tools on the horizon, new ways to edit with even more care, more grace, more wisdom. Let us end then, not with certainty, but with curiosity. In our final chapter, we will peer into the future, not just at what CRISPR has done, but what it is becoming. The story is not finished. The genome is still being read, and somewhere in its strands the next edit is waiting to be written. Sleep comes slowly now, like fog rolling in over still water, and with it, a gentle question rises. Where is this all going? What does the future hold for the language of life we've just begun to read? CRISPR was once a marvel, the tool that showed us we could edit DNA with precision, like a whisper to the genome, correcting what time had marked. But science does not sit still. Even now, new tools emerge from the quiet workshops of biology. They are not louder, they are softer, more precise, more graceful. One is called base editing. Instead of cutting the DNA completely, it simply changes one letter into another, a chemical eraser and pencil in the same hand. No break, no wound, just a subtle correction, like fixing a typo without tearing the page. Another is prime editing, a tool that acts like a word processor for genes. It can insert new sequences, delete old ones, even rewrite short passages without the risks of double-stranded cuts. Imagine it like a tailor, not just mending a thread, but reweaving it so finely that the original flaw disappears without a trace. These tools are still new, still learning, still finding their way through trials, tissues, time. But they promise something once unthinkable, that someday we may edit not just in the lab, not just one cell at a time, but across the whole body. Treating conditions like high cholesterol, not with daily pills, but with a one-time genetic correction that teaches the liver to regulate itself anew. Correcting hearing loss by editing the hair cells in the ear. Restoring muscle strength in those with inherited dystrophies. Even, perhaps, slowing aging by resetting tiny timers inside our cells. All of it possible, none of it guaranteed. The future of genetic medicine is not a promise, it is a path. And like any path, it winds. It requires patience, collaboration, reflection. It requires that we keep asking the right questions, even as the answers change. What should we fix? What should we leave? How do we ensure these tools serve not just the wealthy, but everyone with breath and blood? And above all, how do we remain human in the face of such extraordinary control? Perhaps the answer lies not in power, but in presence. In remembering that the genome is not a machine, but a song. And CRISPR, and all its cousins to come, are simply instruments, tools we use to tune the melody to something more harmonious. The future of medicine may not look like surgery or pills, it may look like editing, quiet, internal, personal. A treatment might begin not with a hospital visit, but with a readout of your unique genome, and from that, a map showing where healing might begin. Your doctor may be part biologist, part bioengineer, part storyteller, helping you understand your code and deciding together whether to gently change a line. And maybe the edits won't come from outside. Maybe your cells will learn to edit themselves. Already, scientists are designing programmable RNA molecules that can drift through the bloodstream, find faulty cells, and quietly repair them, all without surgery, without a single cut. This is called in vivo gene editing, and it holds the promise that the body might one day heal itself from the inside out. These are not science fiction fantasies. They are being tested now in animals, in early trials, in cells that sleep and wake just like yours. And beyond the science, there is still poetry. Because in the end, what we are learning is not how to master life, but how to listen to it more closely. The genome is not a thing to control, it is a story to understand. And as our understanding deepens, our edits become more like conversations, gentle questions to which the body may one day reply. You are part of this story. Your body carries ancient code, but your life writes something new. And while your genome may remain largely untouched, your voice, your choices, will help shape the future of this medicine. A medicine made not only of molecules, but of meaning. So rest now. Let your thoughts curl like strands of DNA, intertwined, mysterious, whole. You have walked through a landscape once hidden from sight, where scissors are made of protein, and healing begins in silence. CRISPR was only the beginning. The edits will continue, the questions will deepen, and somewhere in the double helix a future is waiting to unfold.